Okay, thank you very much. Thanks for the organizer for giving me the opportunity to present the activities. So I won't present the activities of the Institut Merieux, but I will present the activities of the Fondation Merieux because I think that it's more aligned with the topic of the session this morning. So basically, the Fondation Merieux is a non-profit foundation, so that has been recognized of public interest, and its main mission is to help limited resource country to tackle public health issue linked to infectious disease and to improve more globally the living conditions of the most vulnerable. So the foundation has four main objectives: to improve access to diagnostics by strengthening clinical laboratories within national laboratory systems, to enhance local applied research capabilities, in particular within training networks that are coordinated by the Fondation Merieux, to encourage knowledge sharing and public health initiative through the organization of a wide range of international conference at its international center close to the Annecy Lake, and finally to propose integrative and holistic approaches to improve conditions of mothers and children in low-resource countries. So the philosophy of action of the foundation is based on four main components. So collaborative spirit, in the sense that all the projects that are supported by the foundation involve basically most relevant local stakeholders, governmental, private, public, and also international NGOs. A long-term vision, since the foundation has been implementing its project sometimes for more than 15 years, and this sustains support and ensure sustainability of the initiative that has been implemented. A network approach that foster here again international collaboration and experience sharing, which is a key issue, and I will come back to this point afterwards. And finally, the foundation supports global health approaches from human to animals, and including One Health component. So currently the Fondation Merieux is located in around 25 countries, mainly in sub-Saharan Africa. So mainly in sub-Saharan Africa, but also in Southeast Asia. But the foundation is also located in South America, in Haiti, and in the Middle East. So with respect to antimicrobial resistance, so the action implemented by the Fondation Merieux has structured around four main axes. The first one deals with capacity building of laboratory, and I will come back to this point afterwards. The second one focuses on passive and active AMR surveillance. The third one primarily on antibiotic stewardship, and the fourth one on knowledge sharing. And basically, all these activities are very well aligned with at least three of the 5 pillars of the WHO AMR Global Action Plan, focusing on awareness, surveillance and research, and antimicrobial stewardship. So with respect to surveillance, so as you know, the WHO Global AMR Surveillance System, the GLASS, encompass a wide range of activities that are implemented at the surveillance level, at the national level, and at the WHO level. And basically in this organization, naturally the hospital lab and the reference lab play a key role in the process as the main data providers. And basically the Foundation Merieux proposed a multi-pronged approach to strengthen the system and the surveillance, primarily at the surveillance level. So first, by strengthening lab capacity and AMR data collection and representativeness. Second, by strengthening AMR surveillance data quality. And also by fostering integration of clinical data in AMR surveillance. So I will come back to very rapidly to these four main points. So regarding laboratory capacity strengthening, so the Fondation Merieux strengthened laboratory through various ways. So first, the Fondation Merieux can build a laboratory from scratch or refurbish a wide range of laboratory in a variety of countries. And basically, the Foundation Merieux provide also local training of microbiologists and lab technicians, can ensure supply of reagents and equipments. So, as I mentioned, provide adequate infrastructure, can equip them, can allow them to be networked with other laboratories, and also can train the teams for standardized operating procedures. So during this training, we try to focus on possible biases that could be related to laboratory procedures, such as media preparation, inadequate laboratory material, expired consumable, and so on. And also, all these laboratories are equipped with information management system to accelerate data management. So typically, this capacity strengthening is performed in a network approach, because clearly, and I will explain this through the description of one major microbiology network that has been implemented by the Foundation Merieux in Madagascar, which is called RESAMAD. So this is a network of 24 laboratories that has been implemented in collaboration with the Ministry of Health and a number of governmental stakeholders. And basically, the Foundation Merieux is providing technical support for improved diagnosis and surveillance of infectious disease. And the advantage of the network here is that it really fosters, it creates kind of emulation between the various labs. It creates a critical mass of expertise. And through the network, we foster experience sharing, practice harmonization, but also we generally foster pooling of equipment order, external quality assessment, and coordinated training of technicians. In addition, within this network, the Fondation Merieux and the partner ensure that biological activity is not discontinued because of disruption of reagents and consumable. And the Fondation Merieux also help maintenance and acquisition of small material, and has also proposed a cost recovery system for this laboratory in order to ensure their financial sustainability. Regarding the second topic, AMR data representativeness, we have to be aware that in many cases, the trained laboratory are generally located in a tertiary care setting. And basically, so most contributing samples come from patients that have been hospitalized with more severe outcome and comorbidities, and who are not necessarily representative of the whole population, in particular at the community level. And basically, a way to circumvent this problem is to move from a conventional approach for estimation of AMR prevalence to approaches that allow classification of population of settings as having a high or low AMR prevalence, and in this case that require a much smaller sample of size, because typically, for instance, in the case of the lot quality assurance sampling program, which is supported by the European community, typically in this case, the enrollment can be stopped when the required number of resistant isolate has been identified. So typically with this kind of approach, but there are many other ones, you have the possibility to implement much easier population-based rather than hospital-based AMR surveillance. Regarding AMR surveillance data quality, the caveat here is that basically collected AMR data are validated primarily from a microbiological standpoint. And the problem here is that you might have overestimation, for instance, due to colonized samples. You might have problem of missing data, in particular clinical variables, and this was already mentioned by Dr. Poudial. We have also several biases that might be related to data analysis procedures, typically, for instance, because of variability in sample size, but also biases that are linked to prescriber behavior. And so the big issue here is that in low-resource countries, although you can collect AMR data, these data are not validated by an epidemiological experts. And this is a key issue because you can collect tons of data, but if they are not validated formally at the level either of a centralized technical working group or at the level of a national reference lab, then in this case, these data are useless or of limited usefulness. So the priority action of the Foundation Merieux at this level is to train laboratory staff for laboratory procedures and detection of bacterial resistance, which is the basic, but also to strengthen standardization of SOPs for data entry into WHONET to provide consistent data across all Sentinel sites. And what we try to do, which is quite challenging, is to systematically involve an expert epidemiologist to validate the data and to foster then discussion and cross-validation between various focal points, either from a local laboratory, but also from the National Reference Lab or the Ministry of Health, before sending the data to WHO. But this is a key validation step in order to ensure the quality of the data. Regarding integration of clinical data in AMR surveillance, the challenge here is that you have little interaction between microbiologists and clinicians, for the logical reason that in many settings, the quality of microbiology laboratory has been so poor that the clinician has lost trust in biology. And so the big issue here is to restore this bond of trust between the clinicians and the biologist, which is sometimes difficult because the clinicians are not necessarily trained to take properly into account biology report and AST results. And they not even perform biological samples when an infectious syndrome is highly suspected. Irrespective of the issue of the local guidelines, which are often lacking, which are not up to date, or not consulted if existing. So to address this issue, the Foundation Merieux has supported a program called TSARA in Madagascar, which is a real-time AMS pilot program which is based on 3 components: the monitoring of lab results, and clinical and prescription information. So this program involves 11 hospitals, as well as local interns that allow to establish a personalized, bespoke dialogue between the biologist and the antibiotic prescribers, as well as a software that allow integration of national recommendation for antibiotic use, and that allow to collect all the data and to centralize them and to make them available in real time. So basically the objective here is to set up a systematic collection of bacterial ID and AST, as well as clinical data to improve antibiotic prescription and patient care through optimization of empirical treatment recommendation. And so thanks to the software that is made available to the interns, there is the possibility to obtain a real-time collection of AMR data, as well as the trends and impacts of intervention, as well as lab quality assessment, and to inform national stakeholders. And so far, this program has proven to be well uptaken by clinicians, thanks to a personalized approach, and in particular, through this discussion between, of lab data between the prescribers and the biologists. The Foundation Merieux has also been involved in a number of point prevalence survey programs, like the one that has been implemented by the WHO. So basically, the PPS is an approach that is corresponds basically to a snapshot of drug use at a given time or period, which aims at estimating the prevalence of antibiotic use. Typically, a program that has been done by the Foundation Merieux in collaboration with the Fleming Fund in Laos has shown that a PPS performed in several centers in Laos PDR showed that 6 out of 10 patients were under antibiotherapy, that less than 1 in 20 patients had microbiological sample, and that more than 9 out of 10 had a prescription that was not compliant with the guidelines for surgical prophylaxis. So this shows the importance of the problem of antibiotic use in hospital. And basically, based on these results, the Foundation Merieux has launched a number of priority actions that are focusing on training clinicians to send samples to microbiology lab, to take lab results into account when adapting antibiotic therapy, and to consult guidelines for surgical antibiotic prophylaxis. Finally, I would like to mention some One Health approaches that have been implemented by the Foundation Merieux. So basically that tackle the 3 sectors, the environmental, the human, and the animal sectors, as these 3 sectors have been involved as main AMR drivers, as shown by a report published by Holmes and colleagues a while ago. So basically, the One Health approach that has been adopted by the Fondation Merieux, both in Madagascar and in Burkina Faso, correspond actually to the tricycle protocol that has been proposed by the WHO, and which assess AMR prevalence based on analysis of a single reporter bacteria, namely ESBL E. coli. So this is a very easily implementable approach that can be done in a very limited infrastructure setting, and basically the analysis that we have performed about now for 5 years showed a tremendously high level of antibiotic resistance in all 3 sectors. So this program is currently upscaled by integrating additional laboratory in Madagascar, by adding additional reporter bacteria like Carbapenem enterobacteriaceae strains, and by integrating actually whole genome sequencing approaches within the project. In addition, the Foundation Merieux is providing help for several laboratories in Madagascar and Burkina Faso, for instance, to improve their capacity for AMR data collection, processing, analysis, and dissemination, thanks to funding that has been raised through the Gates Foundation. So what are our midterm priorities and challenges? So as already mentioned, surveillance is a cornerstone for understanding the magnitude of AMR, for tailoring policy action, and for tackling AMR. But basically, AMR surveillance data should be reliable, representative, and collected in a systemic manner in order to get relevant information. So basically, the midterm priorities of the Foundation Merieux from a technical standpoint is, as I mentioned before, to keep on improving laboratory capacity, to try to strengthen the links between biological and clinical data, in particular by strengthening this dialogue between the clinician and the biologist, by integrating the One Health approach, which can be challenging, and by strengthening awareness of national health authority. From a clinical standpoint, the objective is that all these surveillance data can have a concrete impact on patient care and outcome. And basically here, the primary objective is more focused on medical indication in order to improve practice at the clinical care level, to generate usable data to improve service to the patients. And finally, naturally, the key objective here is to ensure sustainability of the project over time. So basically, my three take-home message here is it is key to engage all the relevant stakeholders, biologists, clinicians, hospital, relevant local society, ministries, and so on. It's key to ensure the local lab ability to deliver quality microbiological results on time in order to restore this trust between the clinician and the biologist. And so the personalized AMS approach that I just described could contribute to the restoration of this trust between these two stakeholders. Thank you very much.